In this video, we're going to talk about the introduction to basic histology. And what is histology itself? Histology is a branch of biology that studies the tissues. So we said histology study what? Tissues. Which means in histology, what we'll be dealing with are tissues. Now, what are tissues? Tissues are aggregate of cells. You understand? Let me say these are cells that work harmoniously to perform a specific function. So I can say tissues are aggregate of cells. We work together harmoniously to perform a specific function. Remember, in the organization of life, we have something like microorganelles, organelles, cells. But cell is defined as the basic structure and the functional unit of life. So let me say cells. You understand? Now, if you have cells, if you have aggregate of cells, like cells plus cells, we are going to have tissue. Are you getting me now? And if you have tissue plus tissue, we are going to have what? We are going to have organ. If we have organ plus organ, we are going to have system. And we are going to have it like that, like the organization of life. Let me just say cells, aggregate of cells, form tissue, aggregate of tissue forms organs, aggregate of organ form what? Form system, and system now works together to form what? To form a living organism. You understand? A living organism. Now, which means the basic structure and functional unit of life, which is cells, that's the cell, all of them aggregate together to form tissue. Which means in histology, we'll be dealing with cells that comes together to form tissue, as well as the tissue itself, like the tissue that they form itself. You understand? Now, like I said histology is the what? Is the study of what? Is the study of tissue that are arranged to constitute organs, right? I've mentioned that. Now, tissue itself now have two interacting components. Two what? Interacting components. And this component is cells and extracellular matrices. These are the two interacting components that what? That a tissue consists of. Now, the extracellular matrices of many kind of micromolecules, most of them form complex structure, such as what? Such as collagen fibrils. And we'll be talking about all these collagen fibrils, fibroblasts, and other ones when we get to connective tissue. You understand? Now, tissue itself, I said if you're talking about what? If you're talking about histology, we are talking about tissue, right? Now, on these basics, we can now divide tissue into what? Into four. We have four basic tissue in our body, like four of them. We have the epithelial tissue, the connective tissue. We have the nervous tissue, the nervous tissue, and we have the muscles tissue. Now, this epithelia is used as covering, basically. Epithelial tissue is used in covering, and this connective tissue is used in support. This nervous tissue is for control, and this muscle is for movement. So these are the four basic types of tissue that we're having. You understand? Now, what are epithelial tissue? Let me take epithelial tissue first. Epithelial tissue. I'll just introduce you to what we know as epithelial tissues. Epithelial tissue are aggregate of polyhedral cells. Polyhedral cells. The aggregate of what? Polyhedral cells, which means the cells in epithelial tissue are what? Are polyhedral, with a small amount of extracellular matrix, which means they have small amount of what? Of ECM. You understand? They have small amount of extracellular matrix, and the purpose is for protection, lining of surface, lining of surface. We have absorption. They are also used in absorption, and they are also used for what? For secretion as well. Secretion is also part of their function. So they are also used in what? In secretion. Now these are what? This is epithelial tissue. Now let us move to nervous tissue. Now, nervous tissue are tissues that are aggregates of elongated cells, which means in nervous tissue, talking about nervous tissue, we have what? Elongated cells. Elongated cells. And they also have a small amount of extracellular matrix. So they are used for what? They are small amount of what? Of ECM. So I'll be representing extracellular matrix as ECM. So small amount of what? Extracellular matrix. Now, what is their function? They are used for generating and transferring nerve impulse. Generating and what? Transferring nerve impulse. Now, the third one is muscle tissue. Muscle tissue. Now, this one consists of elongated cells as well. But these elongated are not just elongated, they are elongated contractile cells. Contractile cells. Now, they now have a considerable, or let me say moderate amount of ECM. Moderate amount of what? Of extracellular matrix. Are you getting me now? Now, they are used in the condition of movement of body. They are using when body is about to move, you understand? Now, the next one is now connective tissue itself. Connective tissue. Now, connective tissue have fixed and wandering cell. They have what? Fixed and wandering cell. Wandering cell. Now, they now have very abundant ECM. Very what? Very abundant extracellular matrix. That is what they have. And they are used in supporting as well as protecting other tissue. They are used for what? Supporting and also what? Protecting other tissue. Now, what have I mentioned? I mentioned epithelial tissue. Let me use that one to represent E. I mentioned nervous tissue. I mentioned muscular tissue, and I mentioned connective tissue. And I said this extracellular epithelial tissue consists of what? Polyhedral cells with a very small amount, small ECM. Abi? And this one is also made up polyhedral cells, right? And this one is also made up. Nervous tissue is made up of elongated cells. Rather, I made mistake. Elongated cells with a small amount of what? Of extracellular matrix. You understand? This one is made up of what? This one is made up of elongated contractile cells with a moderate ECM. You understand? ECM. But this one is made up of fixed and wandering cell with what? With very abundant ECM. So these are the four tissues that we're having. Now, moving from there, let us move to now. When I said during development of cells and their associated matrix, they become functionally specialized and give rise to fundamental types of tissue with characteristic structural features. Look at it. I said fundamental types of what? Tissue with characteristic structural features. And these form organs. Now, what are the fundamental types of tissue? Those are the four types that I mentioned. The epithelial tissue, the connective tissue, the nervous tissue, and the muscle tissue. You understand? They now have characteristic structural features. And when we get to each of these tissue, I'll be talking about these features of each of them. And one of the features, like few of the features are just what I just mentioned now. They have polyhedral cells. Some of them have elongated cells. Some of them have elongated contractile cells. Some of them have fixed and wandering cells. Some of them have very abundant ECM. Some of them have few abundant ECM. Some of them have abundant ECM and other stuff like that. So when we get to the topic, we are just introducing each other. You understand? We are just introducing each other to histology now. But when we get to the real topic of epithelial tissue, we talk about the special characteristic features that they possess. You understand? Both structural and the functional. You understand? Now, the study of histology now depend on microscope. It depends on what? Microscope. and molecular method of study. Now, it helps in biochemistry, molecular biology, physiology, immunology, pathology, and are essential for better knowledge of tissue. Are you getting me now? Now, basically, now let us talk about the preparation of tissue for study. How do we prepare tissue for study? How do we study tissue? Remember I told you that histology is the study of what? Is the study of tissue. How do we study tissue itself? How do we prepare tissue for study? That is what we are going next. Now, in preparation of tissue for study, it's very simple. We have three. We have three. We have the first one is fixation. Fixation. The second one is sectioning. Sectioning. Sectioning and embedding. Sectioning and embedding. And the third one is staining. So these are the what? These are the three process that we follow in preparing tissue for what? In preparing tissue for study. You understand? So the first one is what? Fixation. The second one is sectioning or embedding. The third one is staining. You understand? Now under this sectioning and embedding, we now have some processes that occur in sectioning and embedding, and they are dehydration, which I'm going to explain in the next slide. Dehydration. The second one is clearing. The third one is infiltration. Infiltration. The third one is embedding itself. Embedding. Are you getting that? The last one is trimming. Trimming. So these are the processes that are under what? That are under sectioning and what? And embedding. Now, let us now talk about each of them. Now, fixation. In order fixation, these are the sequence steps that they follow. The first one is fixation. Fixation, a small piece of tissue is placed in a solution of chemicals that cross-link protein and inactivate degradative enzyme. So the purpose of fixation is to inactivate degradative enzyme so that it won't get spoiled, so that it won't get deformed. Because I want to study a fresh tissue, I don't want it to get deformed. That is the reason why I must fix it first. I must fix it just like the way it is, so that every cells that are present inside, every organelles that are present inside, every tissue that are present inside, they won't degrade. They are not going to be deformed. So I'm using fixation for the purpose of what? Preservation, so that I can preserve the cells and other tissue structures that are inside. You understand? Now, next one is dehydration. Now, under dehydration, the tissue is now transferred through a series of increasingly concentrated alcohol solution. And the purpose of putting it in alcohol solution is to remove water. You understand? From the word dehydration. Removal of what? Removal of water. You understand? That is the purpose of dehydration. Now, after dehydrating it, I must remove the what? I use alcohol in dehydrating it. I must remove the alcohol. You understand? Now, I now use cleaning as a process in removing alcohol. Now, alcohol is removed in an organic solvent. It is removed in what? In an organic solvent in which both alcohol and paraffin are miscible. In which both what? Both alcohol and paraffin is miscible. That is, I'm going to use organic solvent in the removal of what? In the removal of alcohol. Now, next one is infiltration. Now, if it is under infiltration, the tissue is now placed in melted paraffin until it becomes completely infiltrated with the substance. You know what paraffin is? Candle, for example, you can get paraffin from candle. We are now going to melt a candle. A candle, you can melt a candle. Once you melt a candle, you are going to place your tissue that you have removed the alcohol from using organic solvent. You are going to place your tissue inside it. Once you place it inside it, you will now wait until it becomes completely infiltrated. You understand? It becomes completely what? Infiltrated inside the what? Inside the paraffin wax. Now, next one is embedding. Now, the paraffin infiltrated tissue, that is the tissue that you have placed under what? Under the paraffin, is then placed in a small mold with melted paraffin, and we allow it to harden. So that's the purpose of embedding. We allow it to what? We allow it to harden. You understand now? So the next one is now, it has been hardened. once it is hard, what I'm going to do next is to trim it. Now, the resulting paraffin block, that paraffin block is where your tissue is. You understand? The resulting paraffin block is then trimmed so as to expose the tissue for sectioning on a microtome. So we use microtome in sectioning or in cutting. I get me now. We use microtome in what? In sectioning or in cutting. I'm going to tell you what microtome is. Now, similar steps are used in preparing tissue for transmission electron microscopy, except that special fixative and dehydrating solution is used. I get me now. Special fixative and dehydrating solution are used with smaller tissue samples, and embedding involve epoxy resin, not paraffin. We use what? We use epoxy resin when we're dealing with smaller tissues. We don't use paraffin. You understand? Now, let us move to what microtome is. What's a microtome? Microtome is an instrument that is used for sectioning a paraffin embedded tissue for light microscopy. So before you can put something under microscope, a tissue under microscope, what you're just going to do is what? You are going to place it under microtome so that it can cut it. I get me now. And these are the process that which we use in what? In running a microtome. I'm going to show you a picture of a microtome now. So this is a microtome. So look at it. This is a drive wheel. This is the block holder. This is the paraffin block. Look at it here. Look at it here. This is the paraffin block that we place under the microtome. Now this is the tissue that it's section out. You understand? And this is the steel knife that is going to cut the tissue out. You understand? So this is a microtome, and the mechanism of its running is here. So that is how we run a what? This is how we run a microtome. This is the process that which you run a microtome. But we are less concerned about that. Let us just move to what? Let us move to staining directly. Now, most cell and extracellular material, they are completely colorless. So for us to study it under microscope, you have to stain it. Are you getting me now? We now have different type of staining. Dye stained material are more or less selectively, often behaving like acidic or basic compound and forming electrostatic linkage with ionizable radicals of macromolecules in tissues. Now, cells components such as nucleic acid that is having a net negative charge, they have affinity for basic dye and they are termed basophilic. So they have what? Affinity for basic dyes. They are termed what? Basophilic. Why the ones that are having affinity for acidic dyes, they are called what? They are called acidophilic. So we have type of this thing. We have basophilic, we have acidophilic tissues. Acidophilic this thing. Now we now have different types of dyes that we can use in staining. Example of basic dyes includes toluidine blue, Alcian blue, methylene blue, hematoxylin also behave like basic dyes. They stain basophilic tissue as well. Now, the main tissue component that ionize and react with basic dye do so because there is acid in their composition. An example is deoxyribonucleic acid, ribonucleic acid itself, and glycosaminoglycans. Now, example of acidic dyes includes eosin, orangey, and acidic fuchsin. And these are acidic dyes. You understand? They stain acidophilic components such as mitochondria, secretory granules, and collagen fibers. You understand? These are the acidic dye but the most common stain that we use in histology is hematoxylin and eosin. They call it H/E. Hematoxylin, this one, and eosin. That is the most one that we use in what? In histology. But we can use other ones as well, like toluidine blue, Alcian blue. When we get to connective tissue, where we talk about metachromasia, you will see how nucleus undergo metachromasia, how mitochondria and other stuff like that, they undergo metachromasia, where they change the color of the dye to another color. You understand? Like toluidine blue, they can change toluidine blue to brown, to any color, to purple. You understand? That is metachromasia. We'll be talking about their part later. Thank you for listening. God bless you.